Well, imagine that at the social tonight, there's not a choice between a salad or a chocolate mousse, but your menu selection instead comes from the centerpiece. What would you choose? The lilies are full of tannins that will interfere with digestion. The orchids have terpenes that will depress the central nervous system. And the poppies have mind-bending alkaloids. But this problem of food poisoning is one that herbivorous mammals confront at every meal. Approximately 40% of all mammalian species are classified as herbivores, but the majority of mammalian species incorporate some level of plant material into their diet every day. And in turn, plants produce highly bioactive compounds to defend themselves against herbivores and reduce the damage. Let me give you an example of this problem from one of the herbivores on which we've been working, Neotoma stevensii, or Stevens woodrat. Stevens woodrat feeds exclusively on a diet of juniper, and in doing so, ingests a high level of turpentine each and every day. And if we scale up to human body size, the amount of turpentine that stevensii eats would be equivalent to drinking a glass this size of turpentine. Now, since that amount of turpentine is about the lethal dose for humans, you and I would not survive on a diet of juniper. But Stevens woodrat and other herbivorous mammals have been engaged in this arms race with plants for millions of years and have developed curious adaptations and strategies for dealing with dietary toxins. We've been studying a variety of vertebrate species with respect to this problem, and a lot of our work focuses on a single genus, Neotoma, the woodrats. And I'll give you a couple reasons why we've selected this genus to study this problem. The first revolves around the dietary variation in this genus. There are many different species that feed on a variety of toxic plants. There's a wide variation in inter- and intraspecific dietary habits. This genus probably has the greatest number of specialist species within it. And in addition to all this dietary variation, there's an extensive body of literature behind Neotoma. So Neotoma has captured the interest of many of you in the audience. I've listed a few of the people here who have done the preliminary studies that allow us to do the work that we're doing now. So thank you very much for your previous efforts on woodrats. So today I would like to describe 3 general ways that mammalian herbivores deal with dietary toxins. First, they've evolved physiological and biochemical mechanisms to deal with these toxins. Second, they've established collaborations with microbial partners. And third, they've evolved behavioral workarounds to dietary toxins. The first 2 chapters of the story today will be about 15 minutes long, and the last one is a very brief one with a few short videos. So how do herbivores deal with toxins? And what I'd like to focus on for today's talk is how do they actually switch toxic diets? How do they go from feeding on one dietary toxin to a second dietary toxin? So to do that, I need to bring you back to the desert Southwest, shown here, and give you a little background on woodrat diets. The plant juniper is and was a staple food for woodrats. The historical range of juniper, 25,000 years ago, is plotted on this figure in green. The range of the desert woodrat is outlined in dark green, and you can see that juniper and desert woodrats overlap substantially. But we've been taking advantage of a natural experiment that began at the end of the last glacial maximum when the desert Southwest dried out and our deserts were born. And at that time, the plant creosote, shown in hatched bars here, invaded into the desert Southwest. Juniper was extirpated in many areas, and the woodrats that occurred in these regions were forced to switch from a diet of juniper to a diet of creosote. One of the powers of this system is the ability to compare populations of woodrats that currently feed on the ancestral diet of juniper to populations of woodrats that now feed on this novel diet of creosote. And the locations of a couple of our study sites are shown on this map, with the juniper feeders in the triangle and the creosote feeders in the circle. And this evolution of feeding on creosote occurred not just once, but twice. There's another species, Neotoma bryanti, outlined here in blue, that also has populations that occur in the Mojave Desert, and these animals have independently evolved the ability to feed on creosote. It's no small matter to switch from a diet of juniper, of turpentines, to creosote. Juniper produces up to 5% of its biomass in turpentine-like compounds. Creosote produces a very different suite of toxins known as phenolic resins. And the phenolic resins that cover creosote can constitute up to 20% of the biomass of this plant, and it's made up of hundreds of different aromatic compounds. So some of the chemical structures are shown here. When we started this work, we began with a focus on liver enzymes. The liver is the primary site of detoxification in mammals. A complicating factor that I didn't take into account when I first began this work is that each mammalian species produces hundreds of detoxification enzymes. One thing I've learned during this work is one of the reasons why it costs so much to bring a drug to market is that detoxification enzymes are unpredictable. It's hard to estimate from a chemical structure which detoxification enzymes will act on that compound. So we were able to parse out some of this complexity about a decade ago with the advent of microarray technology. This figure displays a microarray where each spot on this slide represents expression of a different gene. When we started this work, we were able to use commercial microarrays that had been designed for lab rats to study detoxification in our Woodrats. But in the subsequent years, we have produced a liver transcriptome for Woodrats that's available to the community, and we've developed customized Woodrat microarrays that focus on liver enzymes. And the design of this microarray is available from Agilent at no extra charge to the cost of the array. So I'm just going to give you two highlights of this work today that lead to our understanding of how Woodrats deal with this diet of creosote. So one thing that we found is that experienced Woodrats that live in the Mojave and feed on creosote, express more detoxification enzymes. And that's displayed in this figure here for Neotoma bryanti. So both experienced populations and naive populations upregulate about the same number of genes when they're fed a diet of creosote. However, the experienced animals express a greater proportion of detoxification genes, suggesting that their detoxification system is ready to handle the incoming toxins. In addition to this, we've found induction of functionally similar genes between Neotoma bryanti and Neotoma lepida. So Neotoma lepida induces aldehyde dehydrogenase in response to feeding on a diet of creosote, whereas bryanti induces or upregulates aldoketoreductase. And while these are different genes and different enzymes, they both function to do the same thing, and that is metabolize aldehydes that have reached the liver. Aldehydes are a common component of creosote resin, and they're also a common intermediate in the detoxification process. And this will come up again in Chapter 2. So in doing all this work with microarrays, we've decided to focus on one particular subfamily of detoxification enzymes, and those are the Cytochromes P450-2B, and I'll just refer to them as CYP2B from here on in. And this is a string and ribbon diagram of CYP2B in the rabbit. We selected to really drill down and work on this family for a variety of reasons. One is it's the largest subfamily of P450s in vertebrates. It's very important in human drug metabolism, and for that reason, a lot of structure-function information is known. There's extensive allelic variation in all of the species that have been studied, and these allelic variations are differences in functions caused by a single nucleotide change. We know that gene duplications are very common in this subfamily. Our work on Woodrats has shown that Woodrats likely have many copies of CYP2B, and at the moment we don't know how many. And our work on Woodrats has revealed population-level differences in the activity of this enzyme without concomitant differences in protein content. So this suggests that different populations have different enzymes that function differently from each other. So we set out to characterize CYP2B, and from here on you'll see these thumbnails of the postdoc or the grad student in my lab that led this research. So Yael set out to characterize CYP2B and went out and collected several individuals of Neotoma lepida from the Great Basin where they feed on juniper and the Mojave where they feed on creosote. We brought them into the lab. We fed them the reciprocal diet of juniper or creosote. And then we set out to sequence the CYP2B in these various populations under these different dietary regimes. And we were shocked with all the variation that we found in these sequences. So when we took the nucleotide sequences and translated them into amino acid sequences, we had 53 different amino acid sequences from these individuals on different diets. And that's summarized in this next diagram that's a maximum likelihood analysis of these different amino acid sequences, where each, you can't see them, but each line represents the amino acid sequence for a particular individual on a particular diet. And the overall pattern that we found here was that there were 4 general clades of this protein, suggesting that there are actually 4 loci within Woodrats that encode for CYP2B enzyme, and that these enzymes are produced in different populations under different dietary regimes. So in general, the pattern was that both populations produce this blue clade of CYP2B enzyme. The Great Basin Woodrats produced this orange clade when they were fed juniper. The Great Basin Woodrats produced the green clade when they were fed creosote, and the Mojave Woodrats produced this pink clade. Well, we further analyzed these data for evidence of selection. And before I go into the results, let me just give you a little bit of detail on how the CYP2B enzyme works. And the inner workings of the enzyme are blown up on this slide here behind me. The gray mesh area represents the active site of the enzyme, and the blue stick represents a toxin molecule within the active site. And the important parts to focus on are these purple sticks here that represent key amino acid residues that recognize the substrate within the active site. So a change in one of these residues changes the function of the molecule. And what we found was that there was a higher rate of change at these substrate recognition sites, these key amino acid residues that change the function of the molecule, non-synonymous substitutions, versus the rest of the molecule, suggesting that there had been selection to change the function of these molecules within Neotoma lepida. We further drilled down to these substrate recognition sites, and there are 7 of them shown here, and that the location along the protein is indicated by the amino acid number along the top here. So there are 7 of these substrate recognition sites that vary among mammalian species. And what we found when we looked at these 4 different clades of enzymes was that the substrate recognition sites had different replacements of amino acids for each one of these 4 clades. So this suggests that each one of these 4 clades functions differently. In addition, the letters in pink indicate novel amino acid substitutions that had not been recognized before our work. So each of these 4 enzymes represents 4 new CYP2B enzymes. We wanted to know if these 4 enzymes really did function differently, so I established a collaboration with Jim Halpert, who's a biochemist at the University of Connecticut in the School of Pharmacy. And Jim has studied this single molecule, CYP2B, in a variety of species for over 30 years. And he looked at our amino acid residues and got very excited and said, absolutely, these enzymes function differently. And these are the homology models. These are what we think these enzymes actually look like that he generated. He also engineered 3 of the 4 of these proteins in his lab and tested them against various pharmacological substrates. And indeed, all 3 of these function differently from each other. After that, we tested these enzymes towards terpenes to see what their affinity was like. And we recorded one of the highest affinities known for a CYP2B enzyme against a substrate. So CYP2B enzymes in woodrats seem to have a very high affinity for terpenes in juniper. And when Jim saw the results of these studies, he said it was as if they were made for each other. We had a long talk about evolution at that point, but he was used to testing enzymes that had no evolutionary history with their substrate because they were pharmaceuticals. So we then went on to compare the affinity of other CYP2B enzymes to terpenes, so CYP2B enzymes from humans, rabbits, rats, and it turns out that this affinity for terpenes is found across these different species. So we're now proposing that perhaps the CYP2B enzyme evolved to deal with dietary terpenes in a variety of species. This work is still really in its infancy. Right now we're developing terpene turnover assays so that we can test our enzymes and other liver enzymes against terpenes directly to see how fast they metabolize these compounds. We're in the process of cloning and sequencing the CYP2B of different juniper specialists. And if you happen to be working on a species that you know consumes a lot of terpenes, I would be happy to take those livers off your hands. So like the cloud rats or the caviomorphs that we heard about this morning. We've been working on sequencing the genome of Neotoma lepida. It's in the final annotation stages, and I hope that resource will be available to the community soon, and it will certainly help us in determining how many different copies of CYP2B woodrats have. All right, so I'd now like to switch gears and talk about how herbivores might get a little help from their friends. The advent of high-throughput sequencing has led us to the appreciation that animals are not individuals, but are complex consortia of microbes, and that these microbes are absolutely essential for health, reproduction, and survival. These microbial partners outnumber our own cells 10-fold, and they outstrip our genome 100-fold. And the amazing thing is, most of this microbial power lies within the gut. Animal scientists have known for decades that ruminants have established complex symbioses with microbial partners that allow them to access the energy in recalcitrant fiber. So we've been asking if all this microbial power in the gut couldn't be harnessed by woodrats to help them detoxify plant secondary compounds. We started by examining the woodrat foregut. Woodrats, like many rodents, have a sacculated stomach with a non-gastric portion shown here in this red box that has a pH of 4.5, which is suitable for microbial growth. And for over a century, scientists have puzzled over the function of this sacculated stomach. And we set out to test whether this section housed bacteria that could detoxify secondary compounds. We found an abundant and diverse microflora in the foreguts of woodrats, and some of the data are shown here. So we have our woodrat gut along the x-axis here and the number of bacterial cells along the y. The foregut housed as many microbial cells as the cecum, and the cecum is a known area that has a high density of microbes with a high microbial activity. We also measured volatile fatty acid production, which is the product of microbial fermentation, and we found that the foregut had VFA production on par with that of the cecum. So we have a high density of cells and a lot of microbial activity, happening in the foregut. We used 16S sequencing to identify the players in this system. We found 39 genera of microbes arranged in 7 different phyla. The predominant phylum was Firmicutes. About 80% of the woodrat gut is made up of members of this phylum. This is in stark contrast to the ruminants that have where Firmicutes make up about 30% of their foregut. So we think that the woodrat foregut functions very differently from a rumen. We also found the phylum Actinobacteria that, combined with the Firmicutes, has members that degrade hydrocarbons. And hydrocarbons are a common backbone structure of plant secondary compounds. So we wondered what the effect of toxins were on microbial function in the foregut. And we did an experiment where we went to the Mojave and collected animals that have experience with creosote. We brought them back into the lab. We put them on a diet that had no creosote, and we put other individuals on a diet that had 2% creosote resin added to rabbit chow. And I should say that 2% creosote resin is a very low concentration of creosote for animals from the Mojave. They can eat far more creosote than this. We then took a metagenomic approach to study the functions, the microbial functions in the foregut, and we classified these functions using MGRAST. This slide displays some of our results of this study with this cluster diagram indicating the microbial communities found in different hosts on different diets. Our control diets clustered together, as did the microbial communities fed to animals consuming creosote. Sorry, that came out a little wrong. Next, what will come up is a heat diagram where microbial genes that are more abundant will be darker in color. So these are the different functions of the microbes in the foregut. We found several of them significantly varied in animals fed creosote versus animals fed control diet. 2 are of particular interest, and that they are the functions of the stress response and metabolism of aromatic compounds. So there were more genes, more microbial genes indicative of these functions in animals fed creosote than control. And this is the same response seen in humans that are taking pharmaceutical compounds. The metabolism of aromatic compounds was of particular interest since creosote contains so many aromatic compounds. And we wanted to drill down and find out which genes were responsible for this difference. And curiously, we only found one microbial gene, aryl alcohol dehydrogenase. This converts aromatic alcohols into aldehydes. It plays a role in the degradation of several organic compounds shown here. And it's not far-fetched to think that it might play a role in some of the compounds in creosote, like NDGA. We found over 50 different copies of this gene in the foregut of woodrats, and that suggests that 50 different microbial OTUs, or 50 different microbial species, are using this gene when the animals are fed creosote. So it suggests that it's very important for the microbial community to have this gene turned on when it's being bathed in a diet of creosote. Now, I said I'd come back to aldehydes from the first chapter. And these results, combined with the data that I talked about in chapter 1, suggest to us that there might be this coevolution between the host detoxification system and the microbial community. And in this example, what we'd like to suggest is that microbes are converting the aromatic alcohols found in creosote into aldehydes, and then the host sees this increased level of aldehydes, and its liver detoxification system has evolved to deal with this high level of aldehydes. So we further asked whether the microbes were critical for consumption of dietary toxins, and we returned to our experienced Neotoma lepida that feeds on creosote, brought them into the lab, put them on 2 diet treatments, control versus 2% creosote, again, a very low dose of creosote. To half of these diet treatments, we added neomycin, which is a broad-spectrum antibiotic that knocks down 90% of the microbes in the gut It is not absorbed by the host, so it does not influence host detoxification in any way. And then we put these animals into a persistence trial to see how long they could maintain themselves on these diet treatments with and without antibiotics. And this slide shows the results of our persistence trials, so days along the x-axis, persistence in the trial before they lost 10% of their body mass on the y. And that flat line across the top represents 2 dietary treatments: the control diet without antibiotics and the 2% creosote diet without antibiotics. So no antibiotics, 100% of the animals were retained during the trial. When antibiotics were added to the control diet, 40% of our animals dropped out by the end of the experiment. We know microbes are important in helping animals to access energy and fiber. However, this is in stark contrast to what happened when we added antibiotics to that 2% creosote resin. None of the animals persisted for 2 weeks of the trial. The microbial diversity in the gut decreased significantly, and we've shown in other studies that we think microbial diversity plays a key role in an animal's ability to deal with toxins. And again, this is a very low dose of creosote resin that these animals were feeding on. So we think microbes are absolutely essential for animals to deal with toxic diets. While we were doing this work, there was a lot of attention being given to new techniques to address disorders of humans. And the technical term being used for this was repopulation of the human gut. So there were these fecal transplants that were taking place between humans to cure various disorders of the gut. And we thought, well, if this can work for humans, surely it can work for coprophagous rodents. So we set out to do a study to see if we could transfer the capability of the microbes into naive hosts and allow those naive hosts to increase their capacity to deal with dietary toxins. So we collected animals from the Great Basin that have no ecological or evolutionary history with creosote, and we separated them into 2 groups, and we collected animals from the Mojave, shown here in green, as well as naive hosts, and we fed our donors a diet of 2% creosote, and we then transferred their feces to the naive recipients. And as I said, Woodrats are coprophagous, and they didn't seem to mind this dietary amendment whatsoever. So we established an experienced transplant group that we validated using 16S sequencing that had the microbial flora of an experienced Mojave animal cast inside a host that was naive. So experienced microflora, naive host. And we conducted another persistence trial to see how these experienced microbes might affect an animal's ability to deal with creosote. The study was slightly different here, where we ramped up the level of creosote to a level of 8% in the diet, which is a challenge for animals with experience. So our Mojave animals tend to start losing body mass when they're feeding on a diet of about 8% creosote. And remember, the animals in this study are all naive to creosote. They have different microflora in their gut. And these are the results. So all of the animals that received the control transplant, which was the microflora from the Great Basin animals, dropped out of the trial by 8% creosote, whereas 40% of those naive animals that had that experienced transplant remained in the study at 8% creosote. So this is on par with what we see in the Mojave Desert. So just by transplanting the microbes of the experienced animals into the naive animals, we've allowed them to feed on a very toxic diet. And we decided to just keep pushing this as far as we could go. We had been studying the microflora of Neotoma albigula, because Neotoma albigula feeds on a diet of cactus, and cactus contains high levels of this compound, oxalate. Oxalate is only degraded by microbes. There is no host contribution to the degradation of oxalate. And we were studying the complex community in Neotoma albigula and its amazing ability to degrade oxalate. We've pushed it up on diets of up to 12% oxalate, and it's still able to degrade most of that oxalate. So we transferred this community across 25 million years of evolution to lab rats, and I'm not going to slog you through more data. I'll just tell you that this microbial transplant from Neotoma albigula increased the oxalate degradation in the lab rat by 3-fold. Now, this may hold promise for humans in the future because oxalate is a common component of the human diet. It occurs in foods like chocolate and rhubarb and spinach. Oxalate is the leading precursor to kidney stones. 1 in every 11 Americans suffers kidney stones, and I know some of you in this room who have told me you've had kidney stones. So it's not too far-fetched to imagine that someday we may be able to humanize woodrat microbes for use as probiotics in humans. So I'm on to chapter 3. This is the final chapter, and this discusses how herbivores have evolved these behavioral workarounds. This is a topic I've been interested in since my PhD studies when I studied this charismatic animal, the North American pika. The pika is an herbivore that lives at high elevations, and it collects food during the summertime to feed on during the wintertime. And these are pie charts of the different diets of pikas at Niwot Ridge. And what I found during these studies was that pikas seem to collect the more toxic plants to store in their diet for the winter. These plants, the toxins in these plants, facilitated preservation, but they decayed over the period of storage. So the animals had more food at the end of the storage period by collecting toxic plants. A few years ago, I had the opportunity to participate in research overseas with 3 Acomes species, or Acomes. Acomes are what the Israelis say, and I realize most people in this room say Acomes. So 3 species of Acomes, Caharinus and Rosatus, both of which live in Israel and feed on this plant, sweet mignonette, shown here. And we added a 3rd species to our study, and this is Acomes minois, which occurs on the island of Crete and was brought there by seafarers 3,000 years ago. So it occurred in North Africa where sweet mignonette occurs, but it was brought to Crete where sweet mignonette does not occur. So this species has evolutionary history with this plant, but no ecological experience. Now, let me tell you a little bit about the fruit of sweet mignonette because it has a very complex toxin system. It promotes seed dispersal by making its fruit toxic only when seed predators feed on it. So when the seeds are crushed, the seeds liberate an enzyme that interacts with the pulp to form the mustard oil bomb, which liberates a series of different toxins. So bona fide seed dispersers can feed on this fruit with no problem, but seed predators get to ingest the toxins in the pulp. And when we started working on these Acomes, we thought that they were all 3 seed predators, but they were not. The golden spiny mouse did what we thought it would do, and it consumes the fruit of sweet mignonette whole, and you can see those black specks in there. They're about the size of sesame seeds. It eats the fruit, seeds and all, and I had gone there to study its physiological adaptations to the mustard oil bomb compounds. But what Mickey, the grad student on the project, noticed was that not all the species did this. That the common spiny mouse had a very tricky way of spitting out the seeds, and one just fell out, so keep your eye on its mouth there, and another seed will be expelled. And this species manipulates the fruit very carefully and spits the seeds out. There's another seed. And this was repeatable. So that's what led to going on to Crete to figure out what this spiny mouse would do with this novel food item that it had evolutionary experience with. And this mouse did something completely different. it carefully extracts the seeds, which are non-toxic by themselves, and discards the pulp. So these 3 mice have 3 different ways of dealing with toxins in their fruit. One somehow tolerates it, one has become a bona fide seed disperser, and one eats the seeds but not the pulp. Well, I hope you have enjoyed hearing about the various ways that mammals cope with dietary toxins. And I'd like to say that we're only at the beginning of a mechanistic understanding of how mammalian herbivores deal with all these toxins in fruit and plants. And I hope that my presentation today will stimulate more research in this area. This basic science not only expands our understanding of sorry, I'm reading and talking at the same time. This basic science not only expands our understanding of the natural world, but it also serves our national interest, which is an important component of NSF grants these days, and holds potential for improving human health and agricultural practices. And with that, I need to thank all the members of my lab over the years that have participated in this work, over 24 creative graduate students and postdocs. And I need to acknowledge my funding sources and my many collaborators, and also the Society for inviting me to give this talk and the amazing award. So thank you very much.